Hi, my name is Karen Miga. I'm from the University of California, Santa Cruz at the Genomics Institute, and I'm also the co-lead of the Telomere-to-Telomere Consortium. And today I'd like to share with you new insights in genome biology and structure that we're learning from our Telomere-to-Telomere chromosome assemblies. Now, as we all know, the human reference genome has been foundational for genomic research. Since its release over 2 decades ago, we've made tremendous progress and almost had a genome revolution, where this particular reference genome is centrally positioned in front of all of our basic science, clinical genetics, as well as now our studies of population genetics, or how we vary between individuals in our population and our genomes sampled throughout our body. However, a common trend of understanding our reference genome is that even though we've been really good at generating lots and lots and lots of human genomes, most, if not all, of the human genomes we've produced so far are incomplete. So there's a real need to complete a human genome. Now, many of you may think, oh, but wait, we had a celebration many years ago about the finished human genome. In fact, we had the release back in 2003, but that focused exclusively on the euchromatic regions and the highly repetitive heterochromatic regions, such as the human centromeres, entire acrocentric short arms, which can represent tens of megabases of sequence, telomeres, subtelomeres, and large regions of segmental duplication were intentionally excluded. And I want to use a slide to just emphasize how important these particular regions are to our understanding of genome biology and function. It's probably very clear to most of the audience that the centromeric regions are important for contributing to also chromosome cohesion and regulating chromosome segregation during mitosis and meiosis. The ribosomal rDNAs, which have been missing from our reference genome, are critically important for life as we know it, as they are important in bringing together amino acids to form particular proteins. And we understand that these regions of the genome have very unique spatial organization, chromatin organization, and these are all parts of our genome that have been not yet explored. And the large unresolved intra and intersegmental duplications could be involved in genome instability and the expansion and contraction of gene families. So overall, I really want to emphasize that what we're missing by having an incomplete genome is interesting and important biology, and we haven't studied it in the past simply because our maps were incomplete. So this is a fundamental problem, and it's a problem that's being addressed by the Telomere-to-Telomere Consortium. This is being co-led by Adam Philippi at NIH and myself, and essentially the goal of this consortium is to organize the international community with one singular goal: to complete the human reference genome, or complete a reference genome of the human genome. And the cell line that we're using is an effectively haploid genome. I want to share what that means with the audience to make sure that you understand it's not a normal genome, in the sense that we're using a complete heterotypical mole. This is a cell line that's derived from an early fertilization accident where we have a complete loss of maternal chromosomes, either before or after fertilization. And then after the entry of the paternal genome, you have a duplication. And so you have the normal karyotype in the sense of having 46 chromosomes. However, if you were to think about it like the image that I'm showing you with the red chromosome, you might have a tandem repeat, which has 20 copies on the maternal side, and a paternal side, which is the blue copy, which only has 10. And that's really complicated to try to sort and assign those particular repeats. However, if you have a duplicated copy, now you have the exact number of repeats on either side, and that really simplifies the assembly problem. Now, all of our reference data that we've been creating, we have a multi-platform approach, which has really emphasized these long read platforms, and all of this is completely open for public use and available on our GitHub. So the challenge here for our consortium was how to do proper repeat assembly. And we're all familiar with the idea of the short read data is very difficult to actually assemble across these highly repetitive regions. Here I'm showing you an example of a 200,000 base pair base pair array of tandem repeats. And if you had short read data, you might have many exact copies within this repetitive region, which could map equally. Therefore, they're of insufficient length to fully span these unique markers that would inform their linear ordering. And as a result, we have collapsed assemblies, and that's what we've been dealing with for many years. Now, we've had a number of long read technologies appear on the scene. One in particular that our consortium started with was the celebration of these ultra-long reads. These are off the Oxford Nanopore platform, where you're able to have reads that are common in the arena of 100 kilobases plus and extending all the way up into megabase space. So this has been tremendous in the idea of traversing gaps in the genome, as previously shown by Jane et al. In Nature Biotech back in 2018, as well as making kind of a targeted completion of human centromeres, as my team showed back in 2018 for the Y centromere. And it was really these two marker papers that inspired us to move forward with the T2T Consortium. And so here you can imagine that if you have that same example of a 200,000 base pair tandem repeat and you have an ultra long sequence, perhaps you can begin to span this entire region completely and then derive the consensus of this entire region based on the spanned repeats. A second type of data that we've been integrating into our T2T consortium that has really had a profound difference on how we're doing repeat assembly is the PacBio high fidelity or HiFi data sets. In this case, you don't benefit from the same read length. In this case, you're looking at more of a 20 kb read. However, each read has Sanger-like quality, where you're looking at Q30. And the way that they achieve that is by actually doing multiple passes of a single template of molecule, as I'm showing you here in a figure. That was taken from the Wegener et al. At Nature Biotech 2019. And you have many error-prone subreads, but if you take this circular consensus, you're able to derive a very high-quality read. And the point here is that this 200,000 base pair tandem repeat that I've been talking about is not a perfect identical copy. There's lots of these smaller perturbations or micro heterogeneity that can differentiate one repeat copy from the other. And without high-quality sequence, you really can't confidently align. But once you do have extremely high quality, like the HiFi platform, you can begin to localize your reads confidently to these unique markers and assemble across. And so our team in particular, giving credit to Sergey Nook from Adam Philippe's group, had been working on HiCanoe. And in this case, what you're able to do is even further improve these already high quality reads. What I'm showing you here, for example, is you can do homopolymer compression, where runs of A, T, Cs, and Gs can be compressed into a single nucleotide to avoid some of the common sequencing errors. You can also do overlap error correction if you find a single nucleotide difference that's not shared with any other read, and ignore some common indels in the tandem repeats. And mainly what I'm wanting you to get from this slide is that you can begin to improve these already stellar reads closer and closer and closer to 100% accurate. And this is really important when we start thinking about centromeric regions, where we have these long chains of tandem repeats or satellite DNAs that are often found from millions of bases. And what we're finding is if you were to look at self-similarity of these centromeric regions, you're really able to capture the heterogeneity and assemble across without doing some multi-mapping or mapping to other parts of the array inappropriately once you get closer and closer to this final polished stage of the HiFi, as I'm showing you here for the self-similarity of 99.999. And so this is exactly what was shown by the HiCanoe paper earlier on, where once you began to improve the quality of even these HiFi reads, we were able to see entire centromeric regions then being assembled. And here I'm showing you chromosome 19 across 5 megabases of repeats. So this is the genome assembly when I started my talk back in 2001. Each one of the oscillations between gray and black, these are breaks in the contig. So you can see that's a highly fragmented genome with many gaps that are still there. And now when we're looking at genome assembly based on our ultra-long data, we're finding that entire chromosome arms are contained in a single contig. And often we are only breaking at these very highly repetitive centromeric regions. And we found that both HiFi and ONT or Oxford Nanopore ultra-long assemblies are highly complementary in the sense that we were finding entire arms being assembled by both technologies, but in certain cases, different chromosomes were better performing in one versus the other. And so it was at that point that we decided to host a workshop. It was a finishing workshop. It was over 100 people joined, and we broke into subgroups. And the goal here was to work on finishing the genome through genome assembly, focusing work on acrocentric short arms, centromeric satellites, segmental duplications, epigenetic profiles, transposable elements, and reference variants. So we have all of these very big and focused working groups that have taken on this challenge to finish the genome. So first, I'd like to go ahead and credit Sergey Nook once again, who was one of the pioneers in some of the early change for us of what's led to this huge development of the release of telomere-to-telomere chromosomes. Essentially, we took a step back and we utilized some of the technology from HiCanoe and the idea that we knew that we could improve the already high-quality HiFi reads. And we could use this information to construct a string graph of long, perfect overlaps. And then we could use the ultra-long nanopore reads to really support the resolution of these harder tangles. What I'm showing you here is kind of all of these string graphs broken into their chromosome assignments, and I hope you can appreciate that each one represents kind of an end-to-end representation of a single chromosome. In implementing this new conservative string graph method that was designed by Sergey Nook, we actually had candidates for T2T predictions for chromosome 11, I'm indicating right away, that came out of our early efforts. We had some clear challenges before us. These are the rDNAs, as I'm trying to illustrate here with the dotted boxes, where we still had links that were being formed across the exact identical regions that were shared across all of the acrocentric regions, and what we're calling the distal junction regions, as well as these long contact links describing a duplication event that took place in the human satellite array on chromosome 9. But apart from that, we did not observe any other interchromosomal links to this assembly, and had a wonderful place to start. And this is just telling the audience, of course, that this is still a work in progress. Our team has been very much focused on completing the rDNA assemblies, but looking at the graphs themselves, I would like everyone to appreciate that the rDNAs actually have sufficient variance to be assigned in a chromosome-specific way, that where we're finding these interchromosomal junctions is really at the level of the distal junction, or these shared regions outside of the rDNA. So the next step for our team was to try to test how accurate these assemblies are, both at the structural level and at the base level. And so to do this, we had to realign all of our data sets to show that we had consistencies in coverage and also base level accuracy. And we did this using two tools. This is WinnowMap, where I'm listing the two publications by Jane et al. At the bottom for folks who are listening to my talk to go back and review and reference, and also something called marker-assisted mapping that we introduced in our Telomere-to-Telomere X paper that came out last year in Nature. And essentially, in the marker-assisted mapping step, you're identifying all of the sites in the genome that have a single copy marker, and you're using these markers to help filter out reads that span unique regions. So in doing this, we were able to accurately align and perform variant calling across the genome. So I'm sharing this page to give, or slide to give folks the feeling of this equal coverage or even coverage that we're observing across. However, the centromeric regions, or the highly satellite regions, were still posing a challenge where we were not seeing completely equal, even and nice coverage across. So this was really passed off to our centromeric satellite working group, where we had a number of really important tools that we onboarded to ensure that these assemblies were corrected where they needed to be and flagged for errors. And this is crediting tandem tools, which I'm showing here. We flag putative errors and then go through and correct on chromosome 10, as well as doing targeted reassembly using orthogonal data sets like ultralong. And this is utilizing technology called Centrifly, where we're able to reassemble using the ultralong reads and show that there's consistency between the high-5 predictions and the ultralong predictions. Additionally, we would look at copy number variation, both looking directly at the Hi-Fi and Illumina PCR-free base data to show that the array links were where we expected them in terms of length and copy number and variant frequency. And we also teamed up with Jennifer Gerten's team to demonstrate that we could use digital droplet PCR to directly target and assess some of these centromeric arrays in CHM13. Overall, we issued a complete sequence of the human genome with an asterisk there because we are still planning to release the rDNA data sets in the near future. But this particular assembly that we've released has 23 chromosomes. This was an XX cell line, so we had no Y, but there are 0 unlocalized, 0 unplaced contigs in this. And overall, we've introduced 100 to 190 megabases of new sequences that were currently not present in HG38. All of our data, our blog posts, and description of these particular sites are present on our GitHub page through our T2T Consortium, and we encourage the use and the open and public use of these data. So some of the big takeaways is that we now have entire acrocentric short arms that we can now study that have never been studied before. This is representing over 90 megabases of newly introduced sequences, where we now have new satellite DNAs, rDNAs, flanking these particular gaps that we're planning to update with the rDNAs themselves, segmental duplications, and hundreds of new candidate gene annotations that we're including in the new reference. just taking kind of a deeper dive into these acrocentric short arms, we're now starting to characterize the satellite patterning that we're observing in the arms and comparing it to the literature to ensure that we're seeing consistency with the fish-based methods, and also diving in even deeper to understand some of these newly discovered sequence features that were missing from repeat masker annotation and previous characterization. Of course, the largest gaps previously in H238 were also in centromeric regions, not only the acrocentric short arms. We knew going into this that centromeric regions were not just a series of tandem repeats, but were kind of a mixture of segmental duplications, many of which could contain paralogous genes, I'm showing you here, as well as interspersed transposable elements. And so our team's been building high-resolution maps of all of the human peri-centromeric regions and centromeric regions, as I'm showing you here. Essentially, the alpha satellite, which is a 171 AT-rich satellite, which is known to be present at every normal human centromere, is absolutely present at all of the human centromeres in CHM13. So we've done a deep dive of characterization of those. In addition, we've been characterizing all of the classical human satellites, beta satellites, and other satellites in this particular cell line as well. And what we're finding is a lot of really cool stuff. For example, we've now noted the discovery of candidate genes and coding regions that now exist in centromeric regions. This is once again crediting the leadership of Mark Deakins, who's been leading our gene annotation team, and Savannah Hoyt, who's been leading some of our transposable element annotation, as well as leading ProSeq, where we're finding examples of gene bodies that exist in centromeric regions that have evidence by ProSeq for having polymerase read-through. Additionally, as I mentioned before, we're doing a huge analysis of the majority of sequences in the centromeric regions. We know that they're defined by collections of satellite DNAs and arrays. And so here I'm showing you chromosome 1, where we have 5 megabases of alpha satellite and also 13.2 megabases of classical human satellites. And so we're able to, for the first time, begin to study arrays of satellite DNA at high resolutions. And so in doing this, we've established a team of experts to go in and begin to provide really clear and well-defined annotation at these particular sites where we can begin to study relationships between the satellites themselves and provide kind of a diction for researchers going forward for how they can begin to describe the satellites in terms of their naming, nomenclature, and organization into repeat structures. One of the interesting things for us, of course, was going into the human satellites 2 and 3. These are less established in the literature as the alpha satellite are. Fortunately, we were able to onboard Nick Altimos, who was a leading researcher in the human satellite arena. He had previously done a genome characterization of these heterochromatic regions involving HSAT2 and HSAT3 and created subfamily characterizations. So we were able to quickly go through and identify that, yes, these predictions match prior characterization of these subfamilies, and that we could begin to study some of the homogenization patterns and repeat structures for the first time. We've also teamed up with a number of repeat experts to start to evaluate with repeat modeler and de novo repeat prediction programs, repeats that may have been missing from H238 that had not been previously described before. This is work that's largely led by Rachel O'Neill's team from UConn. And in this case, we're discovering 208 new repeat predictions, and they map it to roughly 52,000 genomic sites. And we're also finding combinations of transposable elements, or what we're calling composite repeat elements, that are then repeated in tandem over and over again. So these composite satellites are kind of a new discovery for our team, and we're starting to map where they exist in the genome and begin to characterize them as well. This is just another slide to give the audience a feel for these types of composites that we're seeing involving the beta satellite, SST1 satellites, as well as acrocentric satellites as well. So we're starting to see this as a common trend in some of these unknown regions. In addition to the satellites and the acrocentric short arms, we're building comprehensive maps of segmental duplications. We're very lucky to have worked with Evan Eichler and Mitchell Vogel, who are the world's leaders, I would credit, with segmental duplication biology and genomics. And in doing so, we're now able to identify new segmental duplications, as shown in red here, both inter and intrachromosomally. And additionally, to characterizing the presence of these segmental duplications, we're able to quantify the number of segmentally duplicated bases that we're finding outside of HG38, and also focus on the 30 gene-containing duplications where we're seeing expansions and contractions of gene families that had not been well characterized before. So now that we have these incredible maps, how do we use them, as I suggested in my talk title, to actually study genome biology, structure and function? And so to do that, perhaps we need to start moving them into a world where we can start to study not only the genetic organization and structure, but also the epigenetic regulation. This is really, once again, a question of how one can assign reads. For example, if you had an epigenetic data set to these highly repetitive regions. And one method of doing this is to actually anchor these reads using high confident unique markers, or these marker assisted mapping, and that's something that's actively performed by our team. I'd like to start off with the question of studying methylation in these particular regions. This is something that we kind of gained for free using the Oxford Nanopore platform, and this work was led by Ariel Gershman, who is from Winston Temp's group at Johns Hopkins University. Essentially what it is that when you have Nanopore data, you could actually directly probe the chemical structure of the molecule with exquisite sensitivity. So as it's reading through, there's a signal that can be read, and this allows one to begin to predict CpG methylation. But because the reads from the ultralong platform are so long, now we can begin to correlate epigenetic state over hundreds of kilobases of sequence and anchor them confidently to the genome. And so this allows us to build these really comprehensive epigenetic and methylation-based profiles across telomere-to-telomere chromosomes. And each one of the reads, as I'm hoping you can appreciate, originate from a single cell. So not only are we able to look at these methylation patterns in bulk, we're actually able to study the epigenetic heterogeneity within the sample. So as you would expect, there are some basic controls to bring you up to speed. The idea that if you're in a transposable element, you have an expectation to be methylated, and indeed we do find, as I'm trying to illustrate in red, an increase in CpG methylations over the transposable elements. And when you're in a gene body, although you are expected to have methylation, at the site of transcription, you're expecting hypomethylation or unmethylated CpGs, and as shown in blue, that's what we're observing as well. So now if you begin to go into these regions that had not been studied before, such as the DXV4 locus, which is known to be on the long arm of chromosome X, you can then see that this particular site is of interest to many teams because it serves as a tandem repeat that separates pretty large TAD domains on the X. But when you dig in further, we're finding that these repeat structures have a very interesting oscillating pattern of being CpG methylated and CpG unmethylated, as you can hopefully appreciate from this red to blue to red to blue to red to blue. And it really seems to be structured around the organization of the repeats themselves, because once those repeats are inverted and gain some divergence, we see a complete drop-off in CpG methylation that extends out. Perhaps one of the most exciting findings, at least for our team, the Centromeric Working Group, is that we were starting to detect these large hypomethylated regions within each of the centromeric arrays. This I'm showing you here has been published for the chromosome X. That looks to be about 60 kilobases in length. But of course, once we go through all of the new established centromeric regions, we're finding that this trend is true for all human centromeres, that we find this very strategic, but very precise dip that happens across the arrays. So there's a very natural question here when you're thinking about the centromeric regions and you see this type of shift in methylation from CpG methylation to going to unmethylated. And so in this questioning, we had asked, is this actually the site where the kinetochore is being established, or where the functional centromere takes place, where you have the machinery come in and actually touch some of the genomic sites that enable for proper chromosome segregation. And some key proteins here to facilitate that are centromere protein A, as I'm showing in red. So our team next began to ask if we could take a different approach to look at DNA binding proteins to begin to layer more context into these methylation maps. And in doing so, we've teamed up with Gina Caldas from University of Berkeley, who at the time was working with Abby Dernberg, to go through experiments of cut and run. And here I'm trying to show you that, yes, indeed, we identified that the dip coincides with sites of some PA enrichment. This was a finding that was also reported by Glennis Logston, who's recently published a Telomere-to-Telomere 8 paper in Nature. And this hypomethylated region not only correlates with chromosome 8 and chromosome X, as we would expect, but all of the human centromeric arrays that we find this hypomethylated dip, we in fact find that they trend with these sites of kinetochore assembly. So not only do we have now a method established for looking at DNA binding proteins and methylated bases, but we're also interested in looking at RNA transcription that's happening in these particular regions. This is teaming up with Savannah Hoyt, once again from Rachel O'Neill's group at UConn, where we've been anchoring pro-seq reads using this marker-assisted mapping step to study sites of transcription throughout the array. And what we're finding is that the majority of transcription that's happening in centromeric regions are primarily in the transposable elements. We find very, very few reads that are emanating from the satellite DNAs themselves. Additionally, we've been looking at centromeric transcription of the genes that are there to see if any of the particular genes are being transcribed. And we're finding most of the expression in the CHM13 centromeric regions are from the FSHD region gene 1 pseudogenes that are involved in various disease studies. So this is something, too, that we're trying to follow up on to study the differences in the genes that are being expressed in terms of how they're represented in their splice and transcript levels. So overall, what I presented today is a high-resolution characterization of 200 megabases plus of this human genome that's been missing from HG38. And what we're finding are new repeat classes and repeat structures involving inversions and homogenization, and many genes and protein binding motifs that had not been present in our previous assembly. And by having this incredible reference in front of us, we're now able to dive into questions of genome biology and structure by probing DNA methylation, looking at DNA binding, protein binding, and expression profiling. We're not yet to the finish line. Everything that I've showed you today, of course, is using this effectively haploid special line. We do expect there's a technological barrier to getting to diploid T2T genomes. This is something that we've paired up now with the HPRC, or the Human Pangenome Reference Consortium, and we're hoping to reach the production of hundreds of T2T genomes. That's our next barrier that needs to be addressed in the future. And so the hope here is that these new T2T assemblies will really revolutionize genetics and genomics in the sense of having a more complete genome may lead to the discovery of new and important clinical variants that had been overlooked in the past. Additionally, this will expand our understanding of human sequence variation and evolution. And as hopefully I've shared with you over the epigenetic studies, this may lead to new biological insight of how the genome is organized at a cellular level, and how these regions in particular contribute to our understanding of how the cell works. So with that, I'd like to end my talk. This has really been a celebration of collaborative and open science. Our T2T community is open. Anyone is welcome to join. And I would be very happy to take any questions if anyone would like to email me directly. Thank you.